guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about types of enzymes. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So we can classify enzymes into 6 main classes: oxidoreductase, transferase, hydrolase, ligase, isomerase and ligase. So we will talk about them one by one. First is oxidoreductase or dehydrogenase. These enzymes catalyze oxidation reduction reactions between two substrates. So here we have two substrates, X and Y. Now these enzymes catalyze the transfer of electrons from one substrate to another. So we can see that electrons from the substrate Y are transferred to X, like this. And the substrate which donates electrons is called reductant. So Y is reductant here, and the substrate which accepts electrons is called oxidant. So X is the oxidant. Example, lactate dehydrogenase enzyme. This enzyme catalyzes the reversible conversion of pyruvate to lactate with the oxidation of NADH to NAD+ and vice versa. That means this enzyme also catalyzes the conversion of lactate to pyruvate with the reduction of NAD+ to NADH. Next is transferase. These enzymes catalyze the transfer of specific functional group from one substrate to another substrate. So here this is one substrate X and Y is the other substrate. And this A is the specific functional group that is attached to X. And this A is transferred from X to Y like this. So now A is attached to Y here. The example is peptidyl transferase. So during protein translation, amino acid bound to tRNA molecule is transferred to growing polypeptide chain. In this case, this amino acid is the functional group. Next is hydrolase. So these enzymes catalyze the hydrolysis, that is the breakdown of a substrate using water molecule. So here we have the substrate XY and the molecule X and molecule Y, these two are attached. Now this enzyme is breaking down this molecule and X and Y are separated. So it takes the help of water molecule in this breakdown. The example is protease. This enzyme breaks the peptide bonds of proteins. So we can see that glycine and alanine are attached and the protease breaks down by using water molecule and the glycine and alanine are separated here. Next is ligase. These enzymes catalyze breaking of bonds from substrates by mechanisms other than hydrolysis. So here we have the substrate XY and here X and Y, these two molecules are attached. Now this enzyme is breaking down this one and the X and Y are now separated. But this enzyme is not using any water molecule here. So the example is argininosuccinate ligase. Now during urea cycle, this enzyme breaks down argininosuccinate into arginine and succinate. Next is isomerase. These enzymes catalyze the isomerization, that is conversion of a substrate from one isomer to another. Here we have the substrate X and it is getting converted into Y. X and Y are two isomers. The example is phosphoglucose isomerase. Now during glycolysis, this enzyme catalyzes the isomerization of glucose 6-phosphate to fructose 6-phosphate. Next is ligase. These enzymes catalyze the formation of different bonds by combining the two substrates. So here we have substrate X and substrate Y, and this enzyme is combining them into XY. So here X and Y are attached. So the example is DNA ligase. During DNA replication, this enzyme links two pieces of DNA to form a single molecule of DNA. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.